Okay. All right. So we're going to start with kind of general overview. What are microbes? Branches of science that study them, biological macromolecules, role of microbes in our life, and whatnot. So I am a sucker for flowcharts, as you will see. So microorganisms, and I'm gonna do an asterisk because there are some details. What are they? We're gonna start from the smallest ones, which are viruses. Viruses, interestingly enough, are acellular organisms. Okay, so they do not have a cell structure, which doesn't make them living. So they're not dead? I really like the term for them, undead. Okay? They cannot reproduce outside of the host, specifically outside of the host cell, and they are usually observed using electron microscopy. Now, other exceptions, of course, this is why I say usually. Then we've got bacteria and archaea. So these are first cellular microorganisms. They are uniformly unicellular, only one cell. The entire organism is a cell. And they are prokaryotic, meaning that they do not have a nucleus or membrane-bound organ. These microorganisms can be observed with the light microscopy, like the microscope you can see here. But if you want to see, like they can be legitimately observed. Some of them are really small, so you will have to use electron. But again, generally speaking, you start with light microscopy. Then we have fungi. So fungi can be unicellular or multicellular. And they are, sorry, I have all those emails coming over. Just unicellular or multicellular. And they are eukaryotic. Okay? So, like this side, here we go with eukaryotes. Then we have protozoa. By definition, those are unicellular eukaryotes. There are some other features in protozoa that we will address in time. Basically, the main feature, they don't have a cell wall. And here's the reason why I put asterisks next to the microorganisms. It's a group called helminths. Okay? Helminths are always multicellular eukaryotes. And while fungi, well, some fungi you can see with the naked eye, right? Things like mold. Others, like yeast, you have to use light microscopy. Protozoa, for the love of God, dude. It's one student just sending email like every 2 minutes. Light microscopy. Helminths, most of them, not all of them, but most of them, you can see with the naked eye. Okay? The adult organisms, you can see. This is why there is an asterisk. Now, I'm going to put one group with a dashed line here. It's going to be algae. That is one of the few times, well, a couple of times, if not one time in the course, that we mention those. Now, you're all aware of the algae, like big, this kind of grassy things. So they can be multicellular or unicellular. They are eukaryotes. The reason why we do not talk about algae is they do not produce human infection. They do not invade humans. Does that make sense to you or not? Like, you can get allergy from algal bloom, you can get poisoning from algal blooms, but algae will not grow inside of you or on you. We clear? So this is why I do not address algae here at all. Now, I terribly apologize. Okay, questions so far? Straightforward, I think. Last little bit I wanted to add to this is the names of the disciplines that study this. So, virology, love of my life, bacteriology, mycology, elements and protozoa, parasitology. Usually these two are grouped, and mycology. Good? By the way, if any of you in the middle of class wants to take a picture on your phone, I'm fine with it. I'm not saying like do it, it's not mandatory, but I know some students want. Good? Cool. So next story that I want to address is the classification of microorganisms. The branch of biological sciences that deals with the microbial classification is called taxonomy. And it was started by the gentleman named Carl Linnaeus in the end of 18th century. He was the first guy to suggest so-called binomial nomenclature. So let's address this first. Binomial nomenclature means that each name of a living creature, and again, remember, viruses are not living, is the first name is the genus, let's say, Bacillus. Okay, that's the genus name. And then you add a second, and altogether it forms a species name. So, genus, species. Make sense? So, for instance, Bacillus subtilis, Bacillus cereus, Bacillus anthracis, they all belong to the genus Bacillus. They are all different species. And again, species name is two words. Sometimes, if, like to shorten it, if you know what you're talking about, you can just use the first letter of the genus. People, oh yeah, these substance, yeah. Make sense? Viruses. There's no consensus on how to name viruses. Some of them are named based on the taxonomical attribution, what they cause, for instance, papillomavirus, causes papillomas, or SARS-CoV-2. Actually, it's a pretty good name. Not exactly rolling off the tongue, but it's a pretty good name. severe acute respiratory syndrome coronavirus number 2. It's very descriptive. Sometimes viruses are named where they were discovered, like Crimean Congo hemorrhagic fever. Okay? Sometimes they named because of the way they're transmitted and what they cause, tick-borne encephalitis. So it's kind of, a bit of a mess. Okay? Now, obviously, classification should have some kind of relationships between, you have to take all the massive variety of living creatures on Earth and start putting them in different groups based on certain characteristics. So the first such classification was attempted by Whitaker, who divided all the organisms into 5 kingdoms. There's something like this. It's called a phylogenetic tree, more or less. So there was a kingdom Monera. Okay, so those are single-cell prokaryotes. Well, kind of tautology, all prokaryotes. Then there was Protista. So one cell eukaryotic, which was kind of a bit of a, I think it wasn't a great idea because it would put Protozoa and single-celled algae in the same group, and they vastly different. And there were fungi, all of them, including unicellular. And as you know, plantae and animal. Okay? Now, what Whitaker based his classification on? I'm sorry, my mouth was dry, so sometimes take a sip. And if you want to do the same, fine. Just, take it up. See, it's okay. This classification was based on the mostly structural similarities for differences. I'm going to put it structural features. Does that make sense? Like, not to the extent that, dolphin is a fish, but you look at dolphins and whales on one hand and fishes, like, sharks on another, and you clearly see they structurally different, they behaviorally different, they're anatomically different, ecologically different. So Whitaker classification worked fine, but then a guy named Carl Woese got on stage. Carl Woese was a bacteriologist who was studying, he was basically a taxonomist, classifying bacteria. So how you classify bacteria? How you classify, and his approach was different. He worked at the dawn of genomics. So he suggested a very elegant and simple idea. If you take 2 organisms, 2 similar organisms, their genomes are going to be very similar. Like if you take us, our genomes are practically identical. Does that make sense? However, we come from different backgrounds, and there will be some differences in the sequence of our genomes. Make sense, right? Now, where would you expect these changes to? Probably changes will accumulate first in the least important genes, in the least important parts of the genome where changes aren't as critical. Does that make sense? So his idea was if we can identify a very important gene that has to be conserved, that has to be very hard to change, then we can say if these genes between 2 given organisms are identical, then these organisms belong to the same group. If they're just that much different, then they are very close but maybe different species. and the more different they are, the farther the species are from each other. Does that make sense? So the function that he looked at was protein synthesis that is performed by ribosomes. And one of the structural elements of ribosome is a 16S ribosomal RNA in bacteria. So he said that, I'm going to take the sequence of 16S RNA, okay, and I'm going to compare those sequences between different bacteria that I can get my hands on. So far makes sense, right? So he was doing this pretty kind of, I don't want to say menial work, but it's kind of, grindy. And then he borrowed a bacterial species from literally his buddy and colleague down the hall. It was some methanogenic bacteria. And when he compared you would expect a 16S RNA between two bacterial species being ubiquitous. He compared 16S RNA sequence of that new methanogenic bacteria to other bacteria that he already got information from. And that new bacteria is totally different. Like, totally different. Like, another life form different. It's like to discover that your best friend is an alien, So he suggested a different system. And it's really interesting to see how these things in science take off. So he, based on his very thorough genetic analysis, he demonstrated, basically, that there was a common universal ancestor, a protocell, which we all were eventually derived from. And then this common universal ancestor kind of split in terms of the evolution into bacteria, and another branch that contained archaea and eukarya. Now, what you see here is a poorly drawn phylogenetic tree. Phylogenetic. So phyla, different groups, and how they are genetically related to each other. And based on that primitive drawing, you can say that archaea and eukarya, despite the fact that structurally they're very different, are genetically closer to each other than any of these two groups, the bacteria. Basically, these are brother and sister, and this is their cousin. Does that make sense? Now, it's interesting, scientists said, do you think Whittaker's system got dumped? No, they just threw away Monera. A lot of people still use Protista, some people use Protozoa, but everything else, like, look, this is all eukaryotes. All they say, okay, we have domain bacteria, domain archaea, domain eukarya. Inside of domain eukarya, we have protozoa, fungi, animalia, plants. That's it. Kind of synthesized it. We're good? Fantastic. Now, kind of some few trivial things I want to say. First trivial thing, microbes are important. I wouldn't bet, but you can challenge me and give me virtually any biological process, and I will show you how microbes are important in that biological process. So we're talking from decomposition of corpses and dead organic matter to protecting humans from infections by occupying ecological niches in the body, what we call a microbiome. And for human endeavors, drugs, virtually all antibiotics, okay? Bless you. Anti-cancer drugs, some of them were derived from microbes. Food microbiology. Best foods that we have. Come on. Beer, wine, cheese, bread. Basically all liquor is impossible without microbes. Most of the dairy products except milk milk is impossible without microbes because microbes allow cows digest grass. Okay? So practically every food that we put on a table involves microbes some or another instance. Industrial microbiology. Nail polish remover, all these solvents, microbial. Cars that are produced in Brazil, most of them are designed specifically to run on ethanol. Why? Brazil produces a shitload of cane sugar. And then basically they ferment cane and produce a tons of alcohol and throw it in a gas tank. So microbes are extremely important. And only a small fraction of microbes, is actually causing human diseases. To give you, I really like to put this into perspective. My question to you, for you, sorry. So much use of that too. Okay, for you. What do you think in the United States is the microorganism that impacts human health the most? I don't think I know the answer to this question at all. What kind of microbial disease? Any ideas? No? Respiratory infections. Usually lower respiratory tract. So pneumonias, tuberculosis, things like that. If you look at the numbers of the prevalence here, that's probably the most important. If you look at HIV, rates are pretty steady and fairly low. If you look at HCV, hepatitis C, the infection rates kind of go up, but we have a treatment. Unfortunately, vaccine preventable deaths still exist, and they shouldn't. And there are some hospital-acquired infections that take a pretty decent toll. Now, obviously, the elephant in the room is coronavirus. Well, there's a good chance that it will become just another kind of. And is it a dramatic event? Absolutely. And the death toll is huge. What we have kind of lower numbers, 177,000 people, right? And excess mortality, meaning that how many people died over the average for that period, is close to 200,000. It's a lot of people, okay? Now, based on what we know about coronaviruses, it's most likely going to be a common cold eventually. We're going to get population immunity, hopefully by a vaccine. Now I'll give you some perspective on the world impact of some diseases. HIV every year kills 1.7 million people all over the world. Tuberculosis kills about 1.4 million people all over the world. Malaria kills 420,000 people every year, more give or take, and 70% of those deaths are among children between 6 months and 5 years old. Every year. We're not even talking about filamentoses, diseases associated with parasitic worms that are not going to kill you, but they will make your life incredibly miserable, such as hookworm that leads to childhood anemia, impairs the development, including mental development. And these children, they're not disabled, but they are severely disadvantaged, socially and economically. And this is not going to go away, anytime soon or during their lifetime. So the impact of infectious diseases on the society, yes, we measure it in deaths, but we also have to be conscious about, disparities between different countries and differences between, dying and situation when maybe, living with this is much worse than dying, sort of. So we're going to focus in this course entirely on medically important microorganisms. Okay? So in one such microorganism that I want to throw at you first and foremost, I mentioned about microbes that can ferment sugars and produce solvents and alcohols. One of those microbes is Clostridium Acidobutilicum. It produces various solvents, but it's kind of a pariah of its genus, because the first pathogenic genus that we're going to talk about is going to be Clostridium. Now, remember, this is all Latin, okay? So Clostridia is a plural. Like when you talk about someone, like a family, and you say, Smiths, like in plural, Smiths. But then when you talk about a single person, oh, this is John Smith. So when we name a single microorganism, it's going to be Clostridium. Okay? The first one is Clostridium Botulinum, that produces a botulism toxin. It's a neurotoxin, which is, interestingly enough, the deadliest poison that humans know. About, as far as I know, about 4 pounds of this toxin is enough to kill every person in the world. It is extremely toxic. The disease that this and that's important, this is neurotoxin, meaning that bacteria infect humans, right? Oops, the toxin. And toxin causes the disease. So even when bacteria is gone, you're still sick because of the toxin. You don't even need to be infected. You can simply consume the toxin. It's a neurotoxin, causes botulism, and the transmission route for these diseases is mostly in the food. So for instance, poorly clean food, and specifically poorly clean food that is being canned, preserved, may lead to the reproduction of this bacteria. Why? Turns out all Clostridia are found in the environment. They're normal, common environmental bacteria. Okay, nothing wrong with them. They are anaerobes, meaning that they do not like oxygen, but without oxygen, they reproduce just fine, right? Make sense? So when you put Clostridium botulinum in the can, in that anaerobic environment, the food, and you close the can, you put it in the shell. It reproduces there, produces the toxin. Then you open the can, and the flood of oxygen kills all the bacteria. The toxin's fine. You eat the toxin, you're basically done. So what does neurotoxin do? You may remember neuromuscular junctions, okay? Synapses between the nerves and muscles, skeletal muscles. So this neurotoxin, it blocks, I'm going to use this, blocks the release of neuromediator acetylcholine at the neuromuscular junction. There's no vaccine. You can treat it with antitoxin, but it's hard. Botulism is rare, but it's very hard to treat. We good so far? Now, another one that I want to mention is Clostridium tetanus. Now, Clostridium tetanus also produces neurotoxin called danospasmin, and it causes, well, not surprisingly, tetanus or long term. Okay? So, while lack of acetylcholine leads to paralysis of muscles due to inability to contract them, so it's going to be flaccid paralysis. By the way, you die of respiratory arrest. You can't breathe, your diaphragm is completely relaxed. In case of tetanus, which is usually transmitted through the wound, you step on a rusty nail and stuff. It blocks inhibitory upper motor neurons, okay, which leads to spasms and eventually spastic paralysis. Does that make sense? So people with tetanus will exhibit green, clenched fist, flexed elbows, bend backwards, going backwards, all in a spasm, massive spasm. Death also occurs due to the respiratory arrest, because in this case, diaphragm contracts so much that, you cannot breathe. Luckily for everybody, this is a vaccine preventable. It's a component of DTaP vaccine. I've heard about it. There was a case in the state of Washington a couple of years ago when a 10-year-old unvaccinated child got tetanus, and he was cured. It was about 4 or 6 months, and the overall price tag on his treatment was at $810,000. So next time you pay your insurance premiums, think about those more. Okay. How can you get those markers? How can they be in the environment if they are anaerobes? There's oxygen everywhere, right? Well, it turns out not only they anaerobes, they produce a structure that's called endospore. Okay? Now, we'll get to the endospore today. Briefly, endospore, it's not a reproductive structure, not like a fungal spore, okay? This is the special bacterial structure that protects bacterial DNA. It's extremely resilient to environmental challenges, and it can exist in the environment for years. So when you can get endospore that will become a replicating vegetative cell through the wound. Make sense? Clostridium Perfringens. Well, it's called a toxin, but technically speaking, one of many toxins that Clostridium Perfringens produces, e.g., for example, collagenase. Sorry, it's an enzyme that breaks down collagen. It also produces, for instance, so-called alpha toxin that disrupts membranes in the human cells. So now think about it. If collagen, the main component of your connective tissue, is destroyed, your connective tissue essentially falls apart. So the disease that is caused by Clostridium Perfringens is gas gangrene. You've heard about it, I bet, and it affects very much people with diabetes, especially their limbs, especially their lower limbs, because circulation is rather poor and the tissues are hypoxic. And hypoxia for anaerobes is kind of a welcoming environment. Does that make sense to you? Okay, so there you go. There's no vaccine. The main treatment for Clostridium Perfringens is unfortunately amputation. Hyperbaric chambers can help. People are put in a chamber with a lot of oxygen, and that oxygen can remain tissues and basically kill those animals. And last, the thing. Clostridium difficile. CD. A bunch of toxins, mostly toxins that are kill cells, so they lead to ulcerative colitis. So it's an inflammation of a colon that is so severe that may lead to ulcerations and eventually may lead to perforation of the intestine. Perforation of the intestine, if you think about it, leads to peritonitis, and from that point, things only go south. Okay? Make sense? It's a classic example of hospital-acquired infection, and it's a classic example of superinfection that results from intense antibiotic treatment. We have a patient for some other kind of infection, and this patient undergoes long and thorough antibiotic treatment. It may alter this patient's gut microbiome, which protects. When species that represent normal microbiome are mostly destroyed, Clostridium difficile, that may be kind of lurking around, gets an opportunity to replicate in the absence of normal competition. It replicates, it's called superinfection, replicate, and there you go. Why it is a hospital-acquired infection? Because people with severe infections are in hospitals. And Clostridium difficile is kind of, you can test randomly, I think, take 100 people, test them for presence of C. diff. I mean, healthy people, you're going to find some. So it's there, okay? And spores, endospores, make it particularly hard to control in the hospital environment because you cannot bleach the entire hospital. Bleach doesn't do anything. You cannot fill the hospital with hydrogen peroxide without killing the patients, So it's a problem, okay? Now, originally the treatment was kind of, in my opinion, counterintuitive, unleash more antibiotics. But now the more and more common treatment for C. diff is a fecal transplant. Now it's complicated because you need to define what healthy feces are. Originally, feces were collected from donors that were apparently healthy. They were tested for absence of common pathogens. Suspension in the physiological solution was made, and people just drank it. Then they realized that it's not a great idea for many reasons. It doesn't taste good, and it goes to the stomach, which is not good for bacteria. So they started to do enema, which is also not the most pleasant medical procedure. And then now what they do, they freeze dry them, package them in capsules, which have a controlled release. So basically, swallow capsule dissolves in your body. Okay, so that works really, really well. It does work well, and FYI, we've been cleaning as a program for fecal donors, and they've been paid like $40 for donation. I don't know how often you can do it. I know two of my former students, they trying to figure out if they can leave. So that's our Clostridium. Now in a few minutes before we get to a little break, I want to say a few words about where this whole story about, infection and microorganisms came from. The fact that infections may be transmitted was known for a very long time from ancient Greece. Okay? Term quarantine comes from Venice, where teams of ships that were arriving during the outbreak of the, Black Plague. It seems, okay, it kind of auto-focusing. Outbreaks of Black Plague, those ships were quarantined, 40 days quarantine. in Italy. They were quarantined for 40 days in the harbor, Venice, for the one that plague with dog. But what was the biological culprit was not known for a long time because there were some pretty wild hypotheses that living matter can arise from non-living. And in 17th century, I think, Francesco Redi ran the first experiment, which is pretty cool. So you got 3 jars, put their meat, left one jar open, one jar closed with seal, and one jar covered with wax. Just looking whether or not maggots will appear in all 3 jars. As you can imagine, they appeared only in an open one. And he basically demonstrated it's not air, okay? It is flies that have to get in contact with you. Then, in the end of 19th century, Louis Pasteur did his famous experiment with a curved-shaped flask. So it's basically a flask, and the neck is shaped like this. So he filled it with broth, boiled it really, really well, so that here, some of the broth condensed, forming an impenetrable seal. So nothing could get in the flask. And then he left one flask like this and broke neck of another one. Well, guess what? The one with the broken neck, contaminated. This one, as far as I know, is still in the Pasteur Museum in Paris. It's the original one. It's like 125 years old. Still sterile. Okay? Pretty convincing. And then we've got 3 more players. So in medical field, it started with Johannes Zimmelweis, was a physician in the hospital in Vienna. They had a problem with postpartum sepsis. They called it puerperal fever. So women were dying at the rate sometimes reaching 30%. It's pretty chilling. You go into delivery, the doctor tells you, there's 1 out of 3 chance that you're not going to make it. Okay? What he noticed is that women who were cared by regular physicians were less lucky than women who were cared by midwives. And then he figured that physicians would go into the morgue, do biopsies, go back, deliver babies. No gloves, no hand washing. So he instilled a hand washing on them, and the rates dropped like 5%. Okay, so that was pretty. The rest of the story you can Google and read about him on Wiki. It was pretty sad. Eventually, he died in the sun. His colleagues never acknowledged that it was really a great idea. And then there was a guy named John Lister. He was a British surgeon who first developed antiseptic treatment during surgery, used carbolic acid, which is very toxic and very irritating chemical. It's a water solution of phenol. Really, trust me, I worked with phenol in college. It's horrible. So they would spray wounds of the patients. I think that 1860 approximately. And before that, you're just I would just wet the scalpel and start open up, sort of. So no asepsis at all. Okay? So these 2 guys, they kind of proved the concept, because microbes were known, they proved the concept that using antimicrobial techniques, you can reduce the rate of hospital-acquired infections, like post-surgical infections. And then Louis Pasteur suggested the germ theory of disease, saying that, diseases are caused by infectious agents, microorganisms. And Robert Koch, he proposed, was a great guy. I remember cholera and tuberculosis. Those are 2 diseases for which he convincingly demonstrated what causes them, which microbe. But he suggested sort of a methodology how to link the microbial species and the disease. We're going to talk way more about Koch in the infection and immunity, the 4th unit. So far, just know that Koch is he established the method how to link an infectious agent and the disease. So far, does that make sense? We're good. Now I'm going to pause it, and before we go to the break, so we're going to do a conversation about the chemical aspects of blood, biological macromolecules. So first, I want to. So let's talk about biological macromolecules. Call them macromolecules because they will be. Okay. So I want to quickly review what they are and what their functions are. So the first one here will be carbohydrates. Okay. First of all, what are they? We call them sugars, okay? Things like glucose, fructose, galactose, ribose, deoxyribose. There are many more, trehalose, ribinose, mannose. Basically, if there is O's, that's carbohydrate. What do you need to understand? They tend to form polymers. So you have a bunch of elementary building blocks, right? And you link them together. So one building block, let's say a molecule of glucose, is going to be a monosaccharide. And then if you add a monosaccharide and you take it many times and you link them all together, if you link 2, it's a disaccharide. You link 3, it's trisaccharide. You link few, oligosaccharide. And you link many, this polysaccharide. This is the reaction of synthesis that is, as many other synthetic reactions in the cell, is a reaction of dehydration. The result of this reaction, water goes away. And conversely, if you want to bring polysaccharide to individual building blocks, you have to run a process of hydrolysis, which involves water. Does that make sense? So that's going to be synthesis by dehydration, breakdown by hydrolysis. What is the main function? First and foremost, the source of energy, okay? And second, no less important function is a structural function. Polysaccharides are key elements of bacterial cell walls, fungal cell walls, glycocalyx in cells, a lot of structural features. They contribute to the structure of the DNA and RNA, but those two are sort of key. Then we have fats. Well, I shouldn't use the term fat because it's a bit incorrect here. I should use the term lipids, okay? So there are 3 main categories of lipids that I want to discuss with you. One is triglycerides, which consist of a glycerol molecule. Okay, so it's glycerol. In the corners here, the carbons. Okay, and then you have an ester bond with fatty acids. Okay, again, in each corner here is a carbon. So this part right here, it's a glycerol part. This part is fatty acid. So this is why we call them 1, 2, triglyceride. This is why the name. function is energy. Okay? The next one is, or are, phospholipids. So the structure of the phospholipid will be somewhat similar to what you see here, with one little difference. First of all, one of these will be unsaturated fatty acid. And then here, sorry, no. Okay? There's going to be phosphate group. This is why we call it a phospholipid. What's important is that this part of the phospholipid, the glycerol part and the phosphate part, they are hydrophilic. Okay? Hydrophilic. And that part, the fatty part, is hydrophobic, which makes them the perfect building blocks for the membrane. Hydrophilic part is exposed into the watery environment, either outside of the cell or inside of the cell. Does that make sense? While the hydrophobic parts, they form that impermeable hydrophobic barrier between the environment and the cytoplasm. We're going to talk about the structure of the membrane more. Okay, another important feature that will come up, not soon, but at some point. Look at the charges. See negative charges? Cell membrane, whether it's a bacterial membrane or eukaryotic, doesn't matter. It is negatively charged, which is, as it turns out, which is really important for the design of some cleaning agents that destroy bacteria. We good so far? And so the function and structure, namely membranes. And finally, sterols and hopanoids. So sterols and hopanoids, you can look at the structure of those in the lecture notes. They are condensed polycyclic lipids, meaning that there are multiple cyclic structures that are kind of, one lays next to another. Their function is to regulate membrane fluidity or rigidity, whatever term you prefer. What does that mean? Membrane is not a rock wall. Make sense? It is really a fluid. And things in a membrane, such as proteins, for instance, they can move in the membrane, they can float. Okay? But the extent to which they're going to float, the extent to which they're going to move, is determined to a large extent by the structure of the membrane. So sterols and hopanoids can change this membrane structure, facilitating or instead blocking these proteins from moving. They can alter the ability of cell to endocytose or exocytose, like, acquiring stuff or releasing stuff. Does that make sense? What's the difference between the two? Sterols are found in eukaryotes exclusively. Hopanoids found in bacteria. Okay? It's kind of interesting. This is how scientists, for instance, this is how scientists determined when first eukaryotic life forms emerged. It was about 3 billion years ago. They found first traces of sterols in the fossils. I said, well, that's eukaryotic. We good so far? Awesome. Next group is the proteins. So elementary building blocks for the proteins will be amino acids. A typical amino acid looks like this. So this is alpha carbon, which is chemically bonded with the acid and with the amino group, okay? And it also is bonded with the side chain of amino acid. Now, this acid group and amino group can form what is called a peptide bond. Okay? That's a peptide, but this is how amino acids are connected. Okay? Now look at this. We have 21 different kinds of amino acids in our proteins, as well as bacteria and all living organisms. You take these 21 different kinds and you put them in a chain, a certain sequence. Does that make sense? This is what we call a polypeptide, or primary structure of the protein, just the sequence. Then, the side chains, they can be hydrophilic, they can carry a charge, positive charge, negative charge. They can be large or small, and they will start physically interacting with each other. This interaction will lead to folding of the primary structure into a secondary structure, which most common elements of the secondary structure are alpha helices, helical structures, and beta sheets, when it's basically it is like this. So the chain is folded. Okay? But that's not it, because interactions between the amino groups, carboxyl groups, interactions between the side chains make protein folding into a tertiary structure, which is basically a protein globule. Does that make sense here? So think of it as some kind of a magical Lego, that you just build, and then it starts folding by itself. And what you end up with is some kind of a shape, which is tertiary structure. For many proteins, that's it. For some proteins, that's not, and there's several globules, they associate into what's called quaternary structure. For instance, RNA polymerase, bacterial RNA polymerase, it typically consists of five different globules. And each of them by itself is kind of useless, but when they all get together, like Voltron, they start doing things. Okay? Why pay so much attention to the structural features? Number one, essentially, the side chains of amino acids present in the polypeptide will ultimately determine the structure and hence the function of the protein. Does that make sense? And if you manage to change an amino acid, it may lead to change in the structure and the function. Second, you can roll back the whole folding process. It is called denaturation. Okay? And you can go back from denature protein. Essentially, you destroy the structure and you destroy the function. This is what you do when you heat microbes. You denature proteins. Certain chemicals can denature proteins. That denaturation, it deprives proteins from their function, from their structure first of all, and then from their function. This is why the cells die. They just cannot do. Does that make sense? Now, we need to address a couple of really important questions. What are proteins? What do they do? It's going to be a little bit maybe cheesy, but almost everything. What does that mean? Do they form structures? Yes. Do they run reactions as enzymes? Yes. Do they signal in the cell? Yes. Are they causing diseases? Yes. So see, they really, like, proteins do things. 99%, I would say, of cellular function, like function, regular function, like what the cells do on a minute-to-minute basis, is because of the proteins. Okay? Does that make sense? Got it? Fourth group, I'm running out of colors. Nucleic acids. DNA and RNA. Building blocks are nucleotides. Okay? And their role is the storage and transmission, I would say, genetic information. And we will address it more in about 2 minutes. So far, how do you feel about it? Good? Okay, last little bit that is really important for nucleic acids and proteins and carbohydrates, actually. It's a very simple concept of hydrogen bond. Everything that surrounds, well, overwhelming majority, virtually all environments, which we're going to talk about, are watery environments. When microbes infect us, we are giant colloids. We're 60-70% water. Same is true for microbes. So, as a general assumption, there's water outside of the microbe, and there is water inside of the microbe, in the cytoplasm. Does that make sense? So, water is everywhere. So, what you need to remember about water, that it's a polar molecule where oxygen carries slightly negative charge, hydrogen carries slightly positive charge. And this slightly negative can establish a physical bond, not chemical bond, a physical bond with slightly positive. This is why water is liquid at the room temperature. This hydrogen bond is what holds two strands of DNA together. Hydrogen bonds determine the shape of RNA molecules. They determine the shape of protein molecules. Does that make sense? They make glycocalyx formed by carbohydrates outside of bacterial cells. Hydrophilic because carbohydrates are hydrophilic. They can form hydrogen bonds. Does that make sense? Makes sense. Hydrogen bonds must involve hydrogen, but sometimes it can be slightly negatively charged nitrogen, for instance. It can happen, a sulfur. But in this example, we just use oxygen. Negatively charged oxygen, positively charged oxygen. Last bit, very technical, very technical. Concept of pH. I'm going to use this language a lot, talking about changes in the pH. 7 is neutral. Anything above 7 is basic. Anything below 7 is acidic. If pH goes up, the environment is more basic. If it goes down, the environment is more acidic. So if I say, what's going to happen to the cell if pH decreases? You have to process it in the way. If pH decreases, the environment is more acidic, So whatever acid does to the cell. Does that make sense? I understand sometimes it confuses people, but that's how we talk about acidity. Good? Any questions? Stella. Now I'm going to stand on my soapbox and talk about two concepts that I consider the most important concepts that we base our conversations about microorganisms. Concept number one is the concept of gene expression. And you will see that there are two things that will annoy the living shit out of you. First, I love flowcharts. Second, I love analogies. Sometimes my analogies are good, sometimes they are absolutely terrible. I'm more than willing to tell you which one is which. So gene expression, what it is. We can write it in a very simple terms. Some people refer to it as central dogma of molecular biology. DNA, which stores genetic information, gets transcribed into RNA, which is a messenger with the genetic information. And RNA conveys the message to the ribosomes, which do translation that results in the synthesis of proteins. And as you remember, proteins carry out almost all functions in a cell. They basically determine what the cell is like. Does that make sense? Now, the DNA, let's say it's a typical E. coli. The genome of a typical E. coli contains about 5,000 genes. If you would express all 5,000 genes, each gene, presumably, encodes, it's a blueprint for a single protein. If E. coli would produce 5,000 different proteins at every given moment, it is very resource, it has huge resource capacity. You know what I'm doing? Like, it requires a lot of resources. Moreover, it's not required because gene expression changes in response to the changing environment. And here comes my favorite analogy. This time, please don't tell me that it sucks. I will it will really hurt because I love this analogy. Imagine that you have your family recipe book, like the thing that was carried from generation to generation, where every generation of your family, they basically just got down different recipes of different dishes, and they just kind of filed it in this ginormous folder, okay? There are recipes from your parents, your grandparents, great-grandparents. At some point, this is your heritage, right? So you still use it, but it's your heritage. You do not put it in the front lines. You do not put it in a kitchen. You keep it in the living room. That's the end. So you have 5,000 different recipes now, okay? Of all kinds. And your friends call you and tell you, that's changing environment, your friends call you and tell you that they're going to come over for a deal. And you'll understand that there is one person who's celiac, okay? So you have to find something gluten-free. There's one person who's vegetarian, and there is one, Muslim person who doesn't eat pork. I mean, it's not a problem. You just need to make dishes that are suited for that environment, for your guests. Does that make sense? You're not going to cook all 5,000 dishes and put them on the table so they can choose. You're going to just pick some, right, out of this 5,000. But look, you're not going to carry this massive folder into the kitchen to flip through the pages. How would you carry the recipe from your book into the kitchen. You're more than human. Look what you did. Like you have a recipe open, I have a peach cobbler. It's not good. Whatever. What did you do? Huh? It's a look. You have only one of this. You cannot, like, if you spoil it or burn it. How? You and I are not more than humans. I would do the same. But people now would just take a picture with their phone, right? Just take a picture. Well, okay, scribble. Now that picture or that little copy is darn. Now, actually, it's a good example. Let's say you wrote it down on a piece of paper. So you cooked it. What are you going to do with that piece of paper? You'll throw it away. You have original. You don't need it laying around, right? RNA does not hang around for long. It gets degraded really fast in the cell. It's a message. But the result of that message in the kitchen, and ribosomes are in the kitchen, is that you make food, you make proteins. Because your guests, they're not going to eat your recipe book. They're not going to eat photographs on your phone. They're going to eat food. Does that make sense? So think of the gene expression as the fundamental process that defines what the cell is. And cells can change what they are in response to the environment. For instance, E. coli, if you give E. coli lactose as a food, it will start making proteins that can use lactose as a source of energy. Make sense? But if there's no lactose, those proteins are not going to be made. You see? Also, I'm going to introduce the term. I will throw some terms at you from time to time. The term is virulence factor. Anything that determines the virulence, the ability of microorganisms to cause the disease. For instance, flagellum, structure that allows bacteria to move, right? If they're more virulent. They can spread faster. Flagellum is composed of multiple proteins encoded in the DNA. If one bacteria have genes for flagellum, they are more virulent than bacteria that don't have genes for flagellum. Make sense? You can compare E. coli. There are like enteropathogenic E. coli, which causes diarrhea. Okay. And then there is enterohemorrhagic E. coli, 07H157 strain, romaine salad, all that shit. It acquired genes for a specific toxin. And this toxin destroys your colon. Does that make sense? So when you think about the phenotype is not all your genes. Phenotype are the genes that are expressed at the given moment. And it can change. That make sense? Second, really important thing, and actually the coronavirus is a great example for that concept. The concept of evolution, it's a fundamental, probably the most fundamental law for biology as a science. Evolution is the change, right? Adaptive change over time. But we tend to say something has evolved to do blah, 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 okay? Like, if you would look at the structure of the human shoulder and compare it to the structure of shoulders of primates or other mammals, higher mammals, our shoulder is unique. It allows us to throw things. Did we evolve that capacity? No. People who did not throw things, they just didn't survive. They were eaten or they died from hunger. That's it. It's not directional, but also it's not random. Meaning that things just don't pop out of nowhere. And this is a key phrase. There should be some selective pressure. So selective pressure in humans with the shoulder was, food and predators, right? If we would be able to run away from predators and eat on the plant diet, why do we have to throw? Makes sense, right? Why I want to bring up coronavirus and selective pressure. You can see in many news outlets that, oh my God, coronavirus evolves to become more transmissible, less pathogenic, blah, 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 blah. We have no evidence. Okay? You can see that deaths are going down, percentage of deaths, but that's simply because we know how to manage it now. But also about more transmissible, it spreads fast. Well, here's the thing. Is there any selective pressure on the virus to spread fast? No, it's doing exceptionally well in terms of the spread. There's no ecological, evolutionary incentive for it to spread better or to become more or less virulent. Make sense? Another good example of selective pressure, if we want to involve viruses, is herpes versus Ebola virus. If I would offer you a really unfortunate choice between these two, what would you pick? Herpes or Ebola? Be infected by. Really? Herpes, of course. There's nothing wrong with labial herpes. Usually people get it like in a, no, seriously, like I got it when I don't remember, like I was probably in daycare. It's bodily fluids that keeps it spread. Right? So I have cold sores sometimes, and that's it. I'm okay. With Ebola, I have about 10% chance of dying in the United States, being treated here. I don't want it. Here's the deal. Herpes is so tame. If you will inject human herpes into mice will die. But it's so tame in humans because humans and herpes co-evolved for millions of years, literally millions of years. And for a pathogen that requires a host, there's no incentive to kill a host. Does that make sense? If you would, hypothetically, infect this little community with Ebola, well, just one person, and then kind of shut us down, just close the door, give us food, we have two options. Die or not. Many of us will, but there will be no evolution. Eventually, this outbreak will burn out, and the virus will be gone, because it's too bad. It kills too many people. You see what I'm saying? Because those are emerging infections. Coronavirus is an emerging infection. I think we got a pretty good deal on that. It's not that pathogenic. It's not that bad. Could be worse, okay? So, selective pressure on herpes was, okay, I'm gonna kind of become more and more tame so I can spread through the population better and better and better, because every organism, all it needs is to spread. Okay? And for Ebola, it's just accident. All those Ebola outbreaks are accidents. Sometimes pretty large proportions, but accidents. That make sense? So when we think about why some microorganisms behaves in a certain way, gene expression and evolution, antibiotic resistance is evolution, okay? Until you introduce antibiotic in the environment, there is no fitness advantage to be resistant. Does that make sense? Okay. Any questions? We're going to move on and discuss quickly discuss some concepts about the structure of the bacterial cell. There's a page with bacterial cell shapes in the lecture notes. Like round is coccus, rod-like is bacillus and whatnot. No, I'm not gonna, like, it's like just no basic definition. If I ask you a spiral-shaped bacteria, you tell me spirochete or spirilla. Definition, okay? Now, let's talk about bacterial cells. All bacteria have 4 structures that can be found in any cell. First structure is that, remember, there's no nucleus, no nuclei. Nucleoid, DNA. That's the place where genetic information is. Okay? Clear? Yes. Then we have cell membrane. Obviously, cell cannot exist without the cell membrane, which is not the cell. Cytoplasm. We need to fill the cell with something like, gum. And cell needs to make proteins, so ribosomes. Okay? Good? Okay. While we're at it, let's talk about 2 processes. They transport across the membrane and how cells can maintain their shape. So transport. Can be either active or passive. Okay, so passive transport. Chemical is moving from its higher concentration to its lower concentration. This little ball rolling off the slope. We also call this process diffusion. It's basically interchangeable. Does this process require any energy? No, it does not. On the other hand, is active transport, where chemical is moved against its concentration gradient, uphill sort of, and this process requires energy, ATP or any other way. Okay? So far, good? Okay. Now, active transport always requires transmembrane proteins. We call them pumps. Now, in case of passive transport, there are 2 options. Simple diffusion and facilitated diffusion. Simple diffusion, when chemicals diffuse directly across the cell envelope. Does it make sense? They do not need, basically it's like David Copperfield walking through the wall. Okay? They do not need any transmembrane proteins. Molecules that can do simple diffusion are, obviously, they lipid soluble because they have to cross. Remember the membrane, right? You have water-loving heads and water-hating tails. So lipid-soluble molecules can cross that hydrophobic layer in between. Water-soluble molecules, they can't. So if cell has to take in water-soluble molecules, it has to use facilitated diffusion, and those molecules have to enter through channels or carriers. Channels. Okay? Does that make sense? The transport. Why we care so much? Nutrients. Nutrients will diffuse into the cell. Antibiotics. If the cell is flooded with antibiotics, it will pump it out. Make sense? Good? Now, how can cells maintain its shape? It's not a very trivial question. So to understand this, we need to introduce the concept of osmosis. And osmosis is the movement of water, right, across the semipermeable membrane from low solutes to high solutes. It doesn't matter which solute, it's solute. You with me on that? We're good. It may sound a bit counterintuitive, but think of it this way. Water wants to dilute. You see what I'm saying? Water looks for the space where it can dilute further. Got it? So this creates basically three types of environment in which cells can exist. One environment is isotonic, and concentration of solutes inside and outside of the cell is the same. Since there's no difference, there's no high and low, there's no net movement of water. The cell will be just fine. Got it? Then there is hypertonic environment, in which the concentration of solutes outside of the cell is much higher than inside. So where would water go? Bingo. What's going to happen to the cell? It's going to shrivel, and this is how we preserve our food. Why do we salt food? Why can we leave a jam or jelly on the table basically for days? Because salty food, that salt creates very hypertonic environment for microorganisms, and sugar and the jams or jellies creates hypertonic environment for organisms. Make sense? And then there is hypotonic environment. In this environment, concentration of solutes inside the cell is much higher than outside. Well, much. Okay? And in this case, water will move. Excellent. What's going to happen to the cell? Huh? It bursts. It's not going to protect it from bursting, right? Bacterial cells, many of them, fungal cells, algal cells. First of all, majority of microorganisms exist in hypotonic environments, but the cell wall protects them. Makes sense? And this is why bacteria look like, I don't know, like they turgid, like they swell. So, you know? In regards to this, I want to address with you two questions. The first question is this. Remember we mentioned protozoa. Protozoa live in hypotonic environments, like water, like lakes, rivers. And they for sure do not have a cell. So hypotonic environment, water constantly comes in, no cell wall. How do you think they deal with that? Conceptually. Look, it is not a biology question. It is house maintenance question. So you have a house which basically pretty much constantly has a basement that is constantly flooded. Waterproofing is not an option. What's the option? How do you remove water from the basement? I learned about it in Ohio because Louisiana had no basement. Doing some pump, yes. This is what protozoa do. They constantly pump water out. Pumping requires energy. Protozoa always eat. They have to consume food to provide energy for that constant pumping of water. And that actually shows a very interesting thing. Nature is not about optimal, it's about okay. That works. Perfect. Make sense? Second question, which is not as trivial. Now, I'm going to give you a hint. So if you look at the terms hypertonic, isotonic, hypertonic, these terms are relative. You know, I could put numbers not 10 and 100, but 1 and 10, right? It will still be hypertonic. Or 1,000 and 1,000,000, and it would still be hypertonic, right? So now there are bacteria that live in extremely, let's say, salty environments, like Dead Sea. I ask, they live in these extremely, I'm not saying hypertonic, I'm saying extremely salty environments. How can they survive? How are they not shrivel? You live in an environment with hypothetical concentration 1,000, super salty. What do you have to do to not die? See? How? What do they have to do? They have to maintain, think of it. If outside of it is 1,000 and inside is 1,000, it's not a problem, right? So these bacteria, they basically maintain this extremely high osmolality inside of them. If you would put such a bacteria in distilled water, it will blow up. Does that make sense? You see what I'm saying? No? You got it? Just, kind of nod or blink once for yes, twice for no. Okay? Because, look, no facial expressions whatsoever. All right. Questions? Moving on? All right, let's get to some structures that are not in all bacterial cells. Conclusions. Very kind of boring. It's piles of chemicals in the cytoplasm. Okay? Just staying there. Can be nutrients, can be some metals, some inorganic compounds, a lot of different stuff. They're not bound by the membrane, just kind of globules, assemblies of chemicals. All right? Good. Endospore, a pretty interesting structure, which is a survival pod for bacteria. It protects bacterial DNA. You following? Good. So what happens? You have a vegetative cell. Vegetative cell refers to the cell that can reproduce. But then this cell is exposed to some harsh environment. For instance, there's not enough nutrients or not enough water or too many antibiotics or other toxic chemicals. This cell goes through the process called sporulation that results in the formation of endospore. Endospore essentially is a capsule made of proteins and carbohydrates that encases bacterial DNA. It's not a reproductive structure. It's the preservation structure. Does that make sense? Not all the bacteria can make the spores, but those that can, you can imagine the spores are pretty resilient. Once the spores end up in a friendly environment, water, abundance of nutrients, no toxins, they undergo the process of termination, okay, and become vegetative cells. And the spores is a huge issue for microbial control because they're so tough and they can cause the disease. Okay, now we're getting to one of the most important concepts. I'm going to get rid of this if I can find enough space for all the elements here. So this is the cell wall. Not all bacteria have cell wall. Okay? We're going to start with two main types of the cell wall, but before we even get to the types, we need to describe what cell wall consists of. So it has a chains of carbohydrates, okay, which are molecules. You don't need to memorize the acronym. I'm not going to ask for some example. And acetyluronic acid and N-acetylglucosamine. And they know one another. Nam. Does that make sense? You see what I'm saying? One, then another, one, then another. 1, 2, 1, 2. There are multiple chains parallel to each other. But if you try to make a fabric from threads that are aligned in parallel, it's not going to be much of a fabric. So you need to connect those threads, okay, by the linkages, or glycan, which are basically short peptides. I mean, I have only two. They're actually like 12 or 15, I don't remember, right? So what we get here is the glycan chain, glycan because, you know, carbohydrates, glyco, and peptides. So we usually refer to this polymer, all this polymer, as peptidoglycan. The answer to the question, what does bacterial cell wall consist of? You say it consists of peptidoglycan. Chains of carbohydrates connected by linkages formed by multiple amino acids. Why it is important? If you disrupt the synthesis of, let's say, peptides, you weaken the wall and the cell can burst. That's the mechanism of action for penicillin. If the cell fails to form cell wall entirely, for instance, due to not being able to transport building blocks, that's the mechanism of action for basic tracing. Okay, so that's really important to know how this is structured and what do we do with it. Now, there are several types of bacterial cell wall. There are bacteria that do not have cell wall. We'll get to them. But right now, let's address two main types of cell wall, which are gram-positive and gram-negative. We call this way because when you use gram staining, named after Hans Christian Gram, gram-positive, and I'm going to make it even prettier than that, gram-positives, they are stained purple. While gram-negatives stain red. Now, what does gram-positive cell wall look like? So here, you have a membrane, a cytoplasmic membrane. I'm going to call it M, membrane, right? And outside of the membrane, you have a cell wall. Okay? Got it? So we sometimes refer to it as having two-layered envelope. Now, this cell wall contains molecules that are unique to gram-positive microorganisms. Molecules of teichoic acid. Teichoic acid is an example of a virulence factor. It increases adhesion, may trigger specific immune responses in the infected host. That makes sense? And then you have gram-negative cell wall, which of course also has, you have a membrane first. And then you have a pretty thin cell wall. And then you have what's called an outer membrane. So three-layered envelope. Cell membrane, cell wall, outer membrane. In the outer membrane, you can find some interesting molecules, for instance, so-called porins. Porin is basically a channel that allows the transport across the outer membrane. And also, very interesting molecule, which is called lipopolysaccharide, or LPS. Another name for this molecule, not writing like words that you can find in a note, it's called endotoxin. Endo meaning inside. So it's a toxin that is a part of the cell. So far, are you with me? Why I call this molecule very interesting. If you will take gram-negative bacteria, say Salmonella, mix them with 0.9, physiological solution, 0.9% sodium chloride, and then autoclave this thing, you don't have any bacteria anymore, right? No infection can result from, right? You just kill them. Then you take this autoclave, spiked with Salmonella, but already autoclaved solution, and give it to a human. And the human will develop fever, myalgias, arthralgias, headaches, because of LPS. This molecule by itself can cause fever and generalized inflammatory response. Bacteria may be gone, but some symptoms of bacterial infection they actually persist. Does that make sense? This is why we have to be aware of this feature. It's really, really stable, extremely stable. Why do we pay so much attention to gram-positive, gram-negative? As you see, we can distinguish between them based on a particular type of stain. And these differences in the structure also determine the differences in the treatment approaches that we would take towards this bacteria. You have a sore throat, and you go to the physician, they will prescribe antibiotics. Does anyone remember the name? Antibiotic for sore throat. It's amoxicillin. That's like uniform, amoxicillin, right away. Because it's great against the gram-positive bacteria such as strep and staph that cause sore throat. If somebody has a urinary tract infection, they're going to be prescribed some form of cephalosporins of the third generation, ceftriaxone. Why? Because this infection may be caused by both gram-positive, either gram-positive or gram-negative. We don't know. It can be E. coli or it can be Staph saprophyticus. We don't know. Third generation cephalosporin is a broad spectrum that just nuke everything. Okay? If you were bitten by a tick and you develop a rash and a fever and myalgia, it's probably some kind of rickettsial disease. You go to a doctor. What are they going to prescribe? They're going to prescribe doxycycline because it's very effective against gram-negative rickets. So a physician, when they have a patient with unidentified bacterial infection, they're going to send the blood for cultures and blah, blah, blah. But what they really want is just somebody from lab telling them, is it a gram-positive or gram-negative? But then they hold the treatment in onto this particular group of pathogens. Does that make sense? This is why we pay so much attention to the structure of the cell wall. Other options. So, put it this way. There's a mycobacteria, so-called mycolata. They have an interesting structure now. Look. This looks like gram-positive, right? Membrane and cell wall. But outside of that cell wall, they have mycolic acid. It's a waxy fatty acid, here, mycolic acid, which gives them additional layer of protection in human body and makes infections with mycobacteria particularly hard to treat because it allows them to hide inside of macrophages. Like Mycobacterium tuberculosis is very hard to treat. And then there's a whole group of so-called molecules. A couple of examples will be mycoplasma and ureaplasma. You can kind of, remember the word plasma. No cell wall at all. Just no cell wall. So, that makes sense. And, like, seriously, that will define this cell wall structures. They determine how we treat. Walking pneumonia, I guess you're familiar with that diagnosis. Walking pneumonia, ever heard about it? That's mycoplasma infection in the lungs. Can you inhibit the gram-positive cell wall? Nope, it's not going to work. They don't have one. So you need to give some other antibiotics. Z-Pak. Works not even through the cell wall. That makes sense? Good? Okay. Oh shit, sorry, yeah, we done. I was like so excited about this. No, that's great. 10:50, yeah.